All right, let's get started. We are going to finish the second part of the metabolism lecture, um, and we will be drawing more stuff. But first, let's do some quick revision of what we know so far. Right? So far, we know that the cellular respiration is once again C6H12O6, and we will react it with 6 oxygen, and that will get converted into 6 carbon dioxide plus 6 water, and we will produce 36 ATP molecules. That's the main reaction, right? And we call this a redox reaction because some of the reactants, well, the glucose is going to be oxidized while the oxygen is going to be reduced. What that means is we will be taking electrons from glucose, and when we are done taking electrons from glucose, it will be completely broken down into carbon dioxide. Excuse me, so this is reduction. And ultimately, those electrons will end up being on the oxygen. We haven't learned about that part yet, but when it does get on the oxygen, it is going to be, oh, sorry, I wrote it wrong. This is oxidized. Oxidized. Loss of electron, oxidized. And those electrons will ultimately end up on the oxygen, which would be reduced. Reduced into a water molecule. So we know this happens in 4 stages: glycolysis, pyruvate oxidation, Krebs cycle, and the ETC, right? As glucose is being broken down, we can harness energy from it in 2 ways. Okay? So number 1, direct production, or can say that produce ATP directly using substrate level phosphorylation. All right. And secondly, instead of creating ATP directly, we can capture high energy electrons released from glucose. Okay? And then these electrons, as you know, will be used in the ETC to produce ATP through the other method, right? The other method is oxidative phosphorylation. All right. So that's the story so far. By the time we finish Krebs cycle, we have 4 ATPs. Not a lot, okay, just 4. And those 4 ATPs are produced through substrate level phosphorylation. And then we have a bunch of NADH and FADH2, right? So here, produce ATP directly using substrate level phosphorylation. Net, we say 4 net ATP, not a lot. And then capture high energy electron release from glucose, right? Many, we'll count them later. Many NADH, right? Some FADH2. Right? So these are not ATP just yet. They have to go to the ETC. They have to go through some process before we can make the ATP. Now, I want to ask and see if you guys remember. How many ATPs can one NADH produce? Does anybody remember? How many ATP? 3 is correct. Okay, each NADH can produce 3 ATP, whereas each FADH2 can only produce how many? That's correct, 2. All right, let me redirect you to the note we took last time. Right, it's again, you should reorganize the notes if you have to. Right, it says right here, each NADH carries enough energy to produce 3 ATP. Each FADH2 carries enough energy to produce 2 ATP. Okay, I hope that's clear now. So that's where the story ended last time, previously on biology. That's basically the recap. Any quick questions before we move on? All right. So things are about to get even more complicated. And you should probably draw this out. Okay, I would suggest you get a big piece of paper. Okay, like I know you probably just started on your paper, right? But like maybe get another one, turn it sideways, and then you want to draw it like this. Okay, so you're gonna draw this thing, and then you're gonna draw this thing, and then these are things that we're actually gonna draw. Okay, take up the whole paper, and then I'm gonna add things to it. Okay, so there we go. This next part here is called the electron transport chain. All right. I'm not gonna write out everything that I am explaining to you. Whatever I'm telling you, it's actually in the PowerPoint slides, so you can read that up later. But I will try to describe it to you. Okay? But drawing it helps. Right? So this is a mitochondria, right? I hope you recognize that little guy now. Okay? And what we're gonna do is we're gonna zoom in. We're gonna zoom in this little area here. Right? So we have this one, and then we're gonna have this. Okay, I'll give you a moment to draw that. Remember, try to take up the whole page, because we're gonna add a lot of things to it. And I hope you have some colors as well. It will make the diagram easier to see. All right, help me out, please. What is this line? What does that represent? Remember, I'm zooming in onto the mitochondria. So what is this line? What does it represent? No? That's okay. I'll tell you this one. This one is outer mitochondrial membrane. Remember, the mitochondria has two membrane, right? The outer one and the inner one, right? And what does it form? What does the inner membrane form? These, wavy folding. What do we call that?
Cristae.
The cristae, that's correct. Right, and the purpose of the cristae is to increase surface area, right, for reaction. So a couple more things over here, out here. That is the cytoplasm. How about here? What is this thing? The space here.
Matrix.
The matrix, very good, thank you. And lastly, what is that? That's right, the intermembrane space, thank you. All right, so we're basically zooming into the mitochondria, right? Like if this is the mitochondria, we're like basically like going in all the way and then just make it a little bit bigger, right? Any question about the diagram in terms of, where we are and why are the labels the way that they are? Okay, so what is the electron transport chain? Right? What is the electron transport chain? That is basically a group of membrane proteins embedded in the cristae. Okay, so I'm gonna draw some for you. Okay, so that's 1, 2, 3. Okay, 3. That's an ETC. These are proteins. And you might have another ETC here. Okay, you might have another ETC like elsewhere. Okay. So the entire inner membrane is just full of these clusters, groups of ETC. Okay. So that's why you want to fold it up into the cristae. You want to increase the amount of surface area available to put these ETC on, right? Okay, so that's an electron transport chain. That's another one, that's another one. So I'll give you a moment to copy that. Oops, sorry. And we'll continue in a bit. Meanwhile, ask questions if you have any. All right. So along the way of cellular respiration in, again, glycolysis, oxidative phosphorylation, and Krebs cycle, we produce the NADH, right? So they will carry the electrons all the way to the ETC. So there is an NADH. Okay, so the NADH will get converted into NAD again, and then it will let an electron get off. Right? Where will this NAD plus go again? Now that the electron got off at the ETC, where will the NAD plus go? If you're thinking that they will go back to step 1, 2, 3 to get more electrons, that would be correct, right? Remember, step 1, step 2, step 3 keeps on giving out electron from the glucose that they are breaking down, right? So these carrier will go back, get more electrons, and they will come back, drop them off. Go back, get more, come back, drop them off, right? You might have another NADH here, drop off another electron, drop off another electron, right? So this electron is high in energy. We have been calling it high energy electron like the whole time. And the ETC is basically, you can think of it as like stairs, all right? You drop a ball at the top of stairs, and then it just bounces off, right? So this electron is going to get passed down from one protein to the next protein, just like the ball going down the stairs. As the electron passes down the ETC, the proteins here will use the energy from those high energy electron to pump H plus from the matrix into the intermembrane space. Okay, so there are some H plus in the matrix. We're going to pump them into the intermembrane space. Look, I didn't tell you the full story, but it's not too difficult to comprehend here. That NADH, when it becomes NAD plus again, that there is going to be a H plus, right? That's what this H is. This H is the H, right? So we're going to use the energy from the electron, and we're going to pump these H plus all the way into the intermembrane space. Now, I only show you one ETC, but remember there are thousands of these things happening at the same time in the crystal. So over time, you would have high concentration of H plus in the intermembrane space, and you will have low concentration of H plus in the matrix, right? So that's why this is an example of active transport. We are going from area of low concentration to area of high concentration. And instead of powering the transport, the pump with ATP, we are powering it with high energy electron. All right. So let's stop the pump for a second. Let's stop the pump for a second. We're not pumping at the moment. Which way does the H plus want to go? If you let them be and let them do their own thing, which way does H plus want to go? Do they want to go into the matrix on their own, or do they want to go into the intermembrane space? What do you think? What's the natural direction of movement for these hydrogen ions? Well, naturally, things go down their concentration gradient, right? So we have a high concentration of H plus in the intermembrane space and a low concentration in the matrix. So these things would naturally want to go back into the matrix. But they cannot do it because they are charged. And the inner membrane has similar composition as the cell membrane. So it will prevent the charged particle from coming back in. Okay? So you will build up this H plus concentration here. even though they really want to come back in, they have no way of coming back in. So I want you to think about it like this, right? Like there is a waterfall, right? Waterfall coming down, okay? And what happens is we build a wall, we build a dam. Okay? So the dam will prevent the water from falling down, and then the water will build up, right? They want to go down here because they go from high to low area, but because of the dam, right, it's blocking the water. So it keeps on building up, building up, building up, right? And then what we can do is we can create an alternate path, right, for the water to flow through, right? So go through like that. And then you can put a turbine here, a turbine, right? A turbine will turn when the water goes through it, right? And then it generates electricity, right? So instead of letting the water fall through on its own, we damp it and we create an alternate path for it to flow through a turbine so we can harness the energy from the build-up and use that to create electricity. Okay, you don't have to draw this thing. So in a similar sense, instead of building up, a body of water, we are building up a lot of H+, right? They can't come in because of the inner membrane. The inner membrane is like the dam. And then we have an alternate pathway. If there is a protein that looks like a doorknob, okay? And this protein is called an ATPase, or sometimes ATP synthase, same thing. And that's like the turbine in the waterfall analogy, right? The H+ will go through the ATPase. As it goes through the ATPase, it literally turns it, and that allows ADP and P to combine, and then we form ATP. Okay? So ultimately, this process involves two different parts, right? The first part is we gotta create the dam. We gotta create the H+ reservoir. And that part is done by using the electron to pump H+ over. Okay. The second part is to allow the H+ to come back in through the turbine, through the ATPase, so you can convert the potential energy into ATP, convert it into chemical energy into ATP. So that's a lot of information. Please let me know if you have any question. There must be some questions, right? Like if some part is not clear or anything like that, now is a good time to ask.
I just wanted to ask, can you go over the part where you said that it cannot pass through the intermembrane space? Like why do we need the ATP?
ATPase thing?
Yeah.
Okay. So this inner membrane, the inner membrane is same as the cell membrane, right? So it has the phospholipid bilayer, right? Now, the phospholipid bilayer has like a hydrophobic tail in the middle, right? So you might remember things that are charged cannot go through the hydrophobic layer, right? So even though the H+ has a high concentration here and low concentration, even though they want to come back in, they can't just go through the inner membrane like that, okay? Because they are charged, and the membrane will block any charged particle to go through. So the only way to pass through is to go through the ATPase. So we are forcing the H+ to go through this path, And the usefulness of that is that this enzyme, this ATPase, is able to harness the power from the gradient to create the ATP, just like the water dam, right? We got all the water locked up on the top. We let it go through a turbine, and we can use that energy to create electricity. In this case, instead of creating electricity, we're creating chemical energy in the form of ATP. Does that answer your question?
Yes. So does that mean there's a lot of them, the ATPase, like, or
That's right.
just
So that's a good point, right? So like that's not the only guy, right? We might have another one here. We might have another one here. So there are many turbines available. They don't have to go through one single one. You can go through any of them, and any of them will be available to convert the energy into ATP. Okay. Now don't do this, but watch. If we make holes in the inner membrane like that, that's going to be a problem. That's like, the dam that we build has holes in it. So water is just going to be leaking all over the place, right? Instead of going through the turbine. So over here, the H plus is just going to leak through the holes. And that's going to interfere with the ATP production. Okay, remember, this whole process has two parts to it. You need to build up the dam. You need to build up the high concentration of H plus, and you need to let it go through the ATPase. If you interfere with either of these steps, we're not going to be making enough ATP. All right? Any other question? I have a question for you. How many ATPs can we make from these H plus? Okay, so remember, these H plus were pumped over by the ETC using the energy from the high energy electron, right? So how many ATP can we get from that? No? Okay. So the electron was captured and delivered to the ETC by what molecule? By NADH, right? So each NADH carries enough energy to produce 3 ATP. Okay, so the NADH will let the electron off, and then the electron will carry enough energy to pump enough H plus over so that when they come back in, you will make 3 ATP. Okay? Of course, there is the other energy carrier, which is FAD. Okay? FADH2. But FADH2 does not carry as much energy. So instead of depositing the electron on the first protein, it will be depositing it on the second protein. Okay, so it now can only pump, fewer H plus, and that's why it will generate fewer ATP. Okay. I hope that makes sense. You probably have to, listen to it a few more times before it makes sense to you. Now, this is from your PowerPoint. Like I said, you should read it yourself, but I will read it to you once. This is everything I just showed you on the diagram. NADH and FADH2 will deposit the high energy electron on the ETC. Okay, so ETC is just a bunch of proteins in the membrane, right? In the cristae. Where do these NADH and FADH2, where do they come from? They came from the first 3 steps, right? Glycolysis, pyruvate oxidation, and Krebs cycle. Those are the steps where we are oxidizing glucose. We are removing electrons from it. We capture them. That's how we end up with all these things. They drive the electrons to the ETC, and then they put it there. Then it's like a game of hot potato. All right, like this is the electron, okay? Electron is gonna, pass on to the first protein. It's too hot, right? First protein doesn't want it, pass it on to the next one. Next one catches it, pass it to the next one, and so on and so forth. As the electron passes down the ETC, the energy from the electrons are used to pump H plus from the matrix into the intermembrane space. So the concentration of H plus ion increases within the intermembrane space. They want to come back in. Things naturally want to go down their concentration gradient from high to low, but they cannot come back in. Why not? Because they are charged. The H plus ions are charged. Charged particles cannot simply go through a membrane, right? Because of the hydrophobic tails. So we provide an alternate path for them to come back in, right? They have to go through the ATPase, okay, which functions like a turbine. It harnesses the energy from the H plus, much like a turbine harnesses energy from water going through them, and we convert that energy into ATP. That's basically what is happening in this diagram. Questions, comments, anything at all before I tell you more of the story? All right. Now let's switch gear and talk about something else for a second. Sometimes when I take my kids to the park, I get a little bit frustrated. Not at my kids, but then at like other kids on the slide, right? And there's always that kid who goes down the slide and then they stay at the bottom. And then for some reason they don't want to get off. So I always tell my kids, don't slide down if there's someone at the bottom. Okay, you're gonna kick them in the head or something, and that's not nice. You're gonna hurt them, right? So then, my kid would be up there waiting for this person to go away, but they don't go away. And then after a while, the parents would come. Okay, the parents would come, and then they will take the kid away, and then finally the next person can slide down. All right. I'm sure you had that experience before. Now, what does that have to do with anything that we are talking about here? Well, everything is related. Here is the ETC. All right. So the electron got on, and then it get passed down. Okay. So when the electron gets to the end, someone has to come and pull it off the ETC. Right? The parent has to come to get the electron off. If the electron does not get off the ETC, the next electron cannot go. And that's a problem. Okay? If the next electron doesn't go, then the NADH, right, the taxi with the electron in it, gets that ETC. It doesn't know what to do with the passenger. It doesn't know what to do with the electron. The ETC is clogged up, right? There's nowhere to put the electron. And then the whole thing just stops, right? So we need something to pull the electron off. And that something is oxygen. Okay, oxygen. So oxygen is like the parent. Okay, we call that the final electron acceptor. Okay, its job is to pull the electron away, and when it does that, it actually becomes H2O. It will pick up a hydrogen in the matrix as well, and then it will become H2O. So that free up the ETC, so the next electron can go, and you can maintain the electron flow. Okay, that's why we need to breathe. If we don't breathe, we don't have enough oxygen. We don't have enough oxygen. The parent is not there to pull away the kid. The oxygen is not there to pull away the electron. Whole ETC gets clogged up. Electrons not flowing. If the electron is not flowing, we cannot pump H plus. We cannot create the gradient. No gradient, no H plus is flowing through the turbine. No ATP is produced. We die. That's why we have to breathe. Any question about this concept of final electron acceptor? If you think about the overall equation, right? We are ripping electrons off glucose, we're oxidizing it, and then we're capturing all these electrons with NADH, FADH2. But ultimately, after it passes through the ETC, it will end up being on the oxygen, and that's how it becomes the water. So it's not like a direct transfer of electron. The electron actually has to go through many different parts before it ends up on the oxygen. All right, and that's what it is. Now there's another concept that you should know, something called electronegativity. Okay. En. Electronegativity is basically the attraction for electron. High En means high attraction. If something has high electronegativity, it means it absolutely loves electrons. Okay? So the proteins here are organized in a way that the next protein in the series is going to have higher electronegativity. Okay, so the electron hops on, and then the next one loves the electron more. It has a higher En, so it will grab the electron from it, and then the next one likes it more, it will grab it from it, and then the oxygen is the one with the highest electronegativity. So it will be the one that grabs the electron in the very end. Okay, so like in this order, that is gets more and more electronegative. Okay, think of it as like a stronger magnet. Okay, you put a metal ball here, the strong magnet will attract it to the next one, and ultimately onto oxygen. So keep that in mind. We will explain that. We'll use that concept to explain something else in a bit. All right, so let's go back here. All we need to do is add an O2, right? And know that it will become H2O, right? That's all. That's the ETC. That's how we make a majority of the ATP, right? And this particular method of making ATP we call oxidative phosphorylation. Do you have a question, Patricia? I see
Yeah.
yeah,
What
go
is
ahead.
that word next to high?
Highest En, yeah, electronegativity.
Further down at high En.
High En equals high attraction.
Oh, high attraction.
Yeah.
Okay,
So
thank
the
you.
more electronegative it is, the more it is attracted to electrons. Okay.
Okay, thank you.
Yeah. Now, if you have the study guide, please take it out. Someone was sending me a message saying, can we have study guides just like chemistry and math? I don't know what to tell you, that we have been having study guides all along. Yeah, I don't know if you're aware of it. It's all on eCentennial. Okay, so if you do take out your study guide, I want you to go to second page. And if you don't have the study guide, you can, make a similar chart here, right? So this table is going to summarize for you where the 36 ATP molecules came from. Right? One glucose can make 36 ATP, right? Let's take a look at where they come from. So stage one, glycolysis. Glycolysis happens in the cytoplasm, right? You should know that by now. How many ATPs are produced in glycolysis through substrate level phosphorylation? Does anybody remember? We learned that on Tuesday. How many ATPs are produced through substrate level phosphorylation in glycolysis? 4 in total. My tree, that's right. But if we take away the one that we invested, how many net?
2.
2. That's right. So 2 net ATP. Substrate level phosphorylation. This is direct production of ATP off glucose. There's an enzyme that takes a phosphate from the donor and put it onto ADP, right? Direct production. How many energy carrier do we produce? Right? How many NADH do we produce? Again, take a look at the notes we
2.
wrote last time. Sorry, how many, Blessing?
2.
2. Absolutely correct. Now, there is a bit of a problem here. All right. There's always a bit of a problem, isn't there? I'm going to put a star here. Okay. These are produced in the cytoplasm. Okay, these are produced in the cytoplasm. Where do we need to take them? Remind me again. We got these high energy electrons. We gotta drive them all the way to where? Where do they go?
The ETC.
Substrate.
The ETC, that's right. Sorry to interrupt you. The ETC is correct. And where is the ETC? The ETC is in the matrix, isn't it? Okay. So let's take a look at here. Again, you don't have to draw this if you don't want to. You can just, watch. From glycolysis, okay, let me just go like this. So from glycolysis, we produce 2 NADH. Okay, 2 NADH. These 2 NADH needs to go all the way to the ETC, but it's so far away. Like I have to, you're telling me I have to cross 2 membranes to get there, right? Like it's just not possible. Right? The molecule is too big. It can't fit through 2 membranes and get to the ETC. So instead, what happens is, you know what? I will let the electron get off. Right? They can cross the border themselves, and then something else will go pick them up once they cross. Right? So an FAD will come pick them up, and it will become FADH2, and then they will drive them to the ETC. Do you understand? Right? The NADH is too big; it can't fit through, so it will just let the electron off. The electron can go through themselves, and then FAD will pick them up. Is that okay? I am like simplifying things here, right? To just give you the minimum requirement to put everything together. So that, sometimes things might not fully make sense. Yes,
Just
Phoebe.
question.
Yeah.
The FADH will transport the other electrons or the NADH?
The electron, right? So because the so like I guess I should write here. The NADH will let the electron off, and then it will become empty again. So we'll go back and do other stuff and pick up more passengers. But the electron will go through the membrane, and then the FAD will pick it up. Okay, so the electron actually spends some energy going through the membrane. That's why we have a lower energy molecule picking it up, right? Like it's not another NAD that picks it up, right? Because by going through the membrane, it spends some of the energy, right? And so, it's just FAD now instead of another NAD plus. Does that answer your question, or did I make it worse?
You're okay. I'm good. I'm
All right.
okay.
Okay. Anyone else have any other question? Now, I fully understand this is like overwhelming, like so many new things, so many new terms, right? So on the test, I try my best to ask very simple, direct questions, okay, for this particular topic. I'm not going to ask you weird things, obscure things. They should be pretty straightforward. Um, I'm hoping. I'm hoping that you will find it that way. Okay, let's go back to our uh, lovely table here. So, like I said, this is cytoplasm, okay, which is going to become 2FADH in the matrix. I mean, it's not the molecule themselves that becomes FADH2. It's more like they pass the electron. It's like, oh, here, take the electron, please. Okay, I can't go through. You take it. Right? So the electron passes through, and then FADH2 grabs onto it and take it to the ETC. Okay. Justine, so NAD become FAD or NAD pass the electrons. The second thing that you said. Okay. It's just the electron that goes through. All right, not the NAD themselves. Yep. You know, there's this like there are these places in the mall that sells like all kinds of supplements, right? Like you can buy whatever. You can buy vitamin D, you can buy like B12, you can buy like I don't know what else people take, like melatonin and stuff like that, right? Well, guess what? You can actually buy NAD plus and FAD if you want. But you actually don't need to take a lot of them, even if you want to buy them as supplements. You only have to take very little of it, because you reuse them, right? The NADH will become NAD plus, and it becomes NADH. It just keeps on cycling, right? It's not like you're gonna use them up, right? You can also buy supplements. I'm not kidding. Which are part of the ETC, right? Each of these ETC protein has a name, right? One of them is called coenzyme Q10. And next time you go to those supplement store, you can buy Q10. Right? I don't know if it does you any good. Right? You can spend money on it, you can take it, but they are available. Yeah, maybe if you get more energy, you will have more energy. That's right. So back to here. Once we pass the electron through onto the FADH2, right? So we have 2 FADH2. Please tell me you know the answer, all right? For this next part. What do I multiply this by? Times what? What should I times this by? FADH2 times what number to figure out the ATP? Let me go ahead.
3?
But it's FADH2.
2.
2, that's right. Okay, so that's going to be 4 ATP. All right, guys. So these ATP are produced at the ETC using what method, you ask? Well, through oxidative phosphorylation. Right, that's like the dam, right? Building the H plus reservoir, letting it come back in, you know, turning the turbine. So total 4 plus 2, 6 ATP. All right. Next, pyruvate oxidation. That happens in the matrix. How many ATPs are produced through substrate level phosphorylation in step 2? What does the note say? The note from last class. 0 is correct. Very good. 0 ATP. None. Okay, only 3 things happen during pyruvate oxidation, right? You remove a carbon dioxide, you create NADH, and you recharge the molecule by giving it coenzyme A, right? No ATP is produced directly. We do have 2 NADH though. These are already in the matrix, okay? So we don't have to go through those special paths that we talked about, right? You don't need to let the electron off. You can take it directly to the ETC. You're already at the matrix. You don't have to cross 2 barriers, right? So if I take the 2 NADH, once again, what do I multiply by this time? Mhm.
2.
But this is NADH.
NADH is times 2, no?
Let's see what we said last time.
Okay.
It's okay. I'm not sure. I'm sure it's not just you. Like, I look here, right? Guys, get it in your head. Put this sticky tag on your computer screen when you take the test, okay? Each NADH carries enough energy to produce 3 ATP. Each FADH2 carries enough energy to produce 2 ATP. Okay? 3 ATP for NADH, 2 ATP for FADH2. So if we go back to here, because we are talking about NADH, we are going to multiply it by 3. So that gives me 6 ATP, right? In total, we would have 6 plus 0, which is 6 ATP. Okay, I will let you try the last row, please. Okay. Find the notes from last class. Figure out what numbers to put where. Please. And not just the first column, everything, right? In my tree, you got it right for the first column. That's good. Very good. Nice. So this is 2 ATP. If you don't know where these number came from, we actually wrote it down last time. See, from Krebs cycle, we have 4 carbon dioxide, 6 NADH, 2 FADH2, and 2 ATPs, right? Okay. And if you have trouble memorizing these, that's okay. That's why I encourage you to have the cheat note, the help sheet, whatever you want to call it, right? As you're writing the test. So I am gonna ask again one more time. What should I multiply these things by? Okay, 6 NADH times what?
Times 3 and times 2.
That's right, Sarah. Times 3 and times 2, which is 18, right? And 4. Got it? So 18 plus 4 plus 2 gives us 24. Right? Let's add them up. 6 plus 6 plus 24, 36. Okay? Now listen. Of the 36 ATP, 4 are produced by substrate level phosphorylation, right? If you have a calculator, why don't you take 4 divided by 36 and times 100%? What does that give you? That's right, RJ, 11.11%. Let's take a look at what we wrote down last time. Substrate level phosphorylation. Only 11% of ATP molecules are produced this way, right? We just kind of proved it, right? So cool. Should give you goosebumps right here, right? Not a lot of it, just 11%. Not a common way of producing ATP. A majority of the ATPs are produced indirectly, right? As in like not ripping it right off the glucose. It goes through oxidative phosphorylation, creating the damp, letting it pass through the turbine, right? That creates a majority, right? 32 out of the 36 ATP. Okay, that's it. That's the hardest part of the lecture. Right? After the break, we will finish the rest of the lecture, but those are much easier. Right? If you understand everything up to this point, you know, then you should have no trouble with the test. Any final questions before we go on a break? Okay. When we come back, we will go back to the PowerPoint slides. Like I said, all the difficult stuff had already been explained. I don't need to draw any more pictures. We have about 20 more slides to go. So if you don't have the PowerPoint, please go download that. And if you think of any question while we are on break, please feel free to type them out. Okay, so I'll see you in about 5 minutes. Okay, 5 minutes, that is what, 1:34-ish. Yeah. See you in a bit. So today, right? Like the lesson so far from today is pretty much talking about oxidative phosphorylation, right? There are 2 parts to it: generating the gradient and letting it flow through the turbine. If you interfere with either part, you are not going to be able to create ATP, and you might die from it. So there are a couple of poison that we're going to look at, and these poisons will interfere with different parts of oxidative phosphorylation. So first of all, they can interfere with oxidative phosphorylation by blocking the ETC. Right? So if it clogs up the ETC, electrons can flow, and if you don't allow electrons to flow, you cannot pump H+, no H+, no ATP. Inhibit the turbine, you cannot harness the energy from the H+, you cannot create ATP. Last one, it makes the membrane leaky. Right? So instead of flowing through the turbine, they just leak back into the matrix, not creating energy in the process. So let's take a closer look at some of these poison. Rotenone. Okay, so that is used as an insecticide and pesticide. Of course, it's relatively effective because insects and pests, they are also animals and they also produce ATP. So if you use this to disrupt their ATP production, you will kill them. But it's not too safe if it makes themselves into the food chain, right? If the crops are not washed properly, if it gets into the birds and so on and so forth, it could end up in a food sources, and that of course is not a good thing. So what it does is it binds to the first protein in the ETC and it inhibits it. Right? Essentially, it blocks the first protein here. So the NADH comes, but it doesn't know where to put the electron. Right? It's like the ETC is closed. They don't know where to put the electron. Right? So if you cannot deposit the electron, there's no electron flow. No electron flow, no H+. No H plus, no ATP. We die. Carbon monoxide. Right? You should have a carbon monoxide detector at home. It's by law, everyone should have one because you cannot smell it, you cannot taste it, and you cannot see it. Right? So how carbon dioxide is poisonous is that it will bind to hemoglobin and it will displace oxygen. Hemoglobin is the protein that helps you transport oxygen in the body, right? But when given a choice between carbon monoxide or oxygen, it will always choose carbon monoxide over oxygen. It's like my kids. Do you want chicken nuggets or a salad for dinner? Always the chicken nugget. Chicken nugget is the carbon monoxide. Okay. But carbon monoxide cannot pull electrons from the ETC. Right? We need the oxygen to do it. So without the oxygen, right, your ETC clogs up. No flow of electron, no hydrogen, no ATP. We die. Okay. Cyanide. Cyanide. What is the formula for cyanide? You learned that in chemistry, right? Anybody remember?
CN.
CN, that's right, CN negative, right? So CN negative is even more electronegative than the oxygen, right? So it binds to the protein in the ETC and basically clogs up the chain again, right? All these things ends up clogging the ETC. Right? So look here, I was telling you that the oxygen is the one with the highest En. It's able to pull the electron off the ETC, right? So that you maintain the flow. But if you have cyanide, cyanide will come and cyanide has even higher electronegativity than the oxygen. So in the presence of cyanide, the electron will not even want to get off. It's stuck to the chain. It's stuck to the ETC. Okay? You can't pull it off. And then for the last time, if the ETC clogs up, you don't pump H plus. If you don't pump H plus over, you don't have a gradient. You don't produce ATP. DNP. Okay? This thing was discovered, accidentally. Right? There was it was first like discovered accidentally. Okay, let me rephrase that. This was used in a factory for some other purposes. Okay, I think it was like for like stabilizing paint or something. I don't remember exactly. Okay, but then you were not supposed to eat them. But then the worker who was working in the factory at the time accidentally consumed some on the job. Right, and then he experienced unexpected weight loss, like very quickly. Within a week, he lost a lot of weight, right? And then after people find out about it, they thought they hit the jackpot for weight loss drug, okay, for anti-obesity therapy, for example, right? And the pharmaceutical company actually jumped on that and marketed it as a weight loss compound in 1934. Okay, but they didn't really fully understand the how it works. Well, how it actually works is that it will transport H plus across the inner mitochondrial membrane. So the first set of poison we talk about prevents us from creating the H plus gradient, right? Prevents us from building up the dam, basically. But this drug here prevents the flow of H plus through the turbine. It blocks it from going through the turbine. Instead, let me show you the picture here. In the presence of DNP basically will be like, hey, you know what? Let's not go through the turbine, okay? Go through me instead, right? So they'll move the H plus over. But of course, the DNP is not creating any ATP for you, right? So when someone consumes DNP, they are not making ATP. They're not making as many ATP, okay? Because not all of them are going through the turbine. And the body thinks you are starving. So your body tries to compensate by increasing metabolism. They try to burn off the fat, and that's why you lose weight rapidly. But burning a lot of fat in a very short period of time is dangerous. It actually causes your blood pH to drop. And it turns out the DNP will also increase your heart rate and kind of mess up your whole metabolism in the body. And people actually could die from consumption of DNP. And people did die as a result of that. So they pulled the plug on that one, is no longer sold as a weight loss drug. But in the dark corners of the internet, some people can still get their hand on it, right? And several people actually still die in the U.S. as a result of consuming tablets that contains DNP. So, victims, parents are trying to advocate for tighter control, tighter regulation, and a crackdown on these illegal sales of these very dangerous compounds. Okay, so this is just summary, right? So rotenone, cyanide, carbon monoxide, they all clog up the ETC. DNP just bypassed the turbine completely. And there's something called oligomycin, which was the second one in the earlier PowerPoint slides. It blocks the turbine, this particular one. Okay, so all these are very, very poisonous, right? If you stop ATP production, you can't do anything. Your heart will not be able to beat without ATP, right? That's why it's poisonous. That's the chart that we wrote before the break. You can take a look again when you have time. We don't live on carbohydrates alone, right? In fact, you probably have heard if your diet is just carbohydrates, is probably going to be not very healthy for you, right? So we eat other things, right? Proteins, lipids, and these molecules can actually be broken down and be fed as intermediates in various parts of cellular respiration. So for example, the proteins, depending on which amino acid it is, some amino acid can go into the citric acid cycle directly. Some will be converted into acetyl-CoA. Some will be converted into one of the intermediates in glycolysis. Remember, glycolysis is actually 10 steps, right? So each step is the molecule, and some amino acid can be converted and feed into those pathways, right? Same thing for fats, right? So we are quite versatile when it comes to the kind of fuel that we can burn, although sugar, like glucose, is the one that is the preferred source of energy. We are capable of breaking down other stuff as well. Okay, this question is going to be on your test. Why don't you read it and tell me what you think? Feel free to type the answer in the chat box if you want, or just unmute and tell me. iPhone 12 says B. Anyone else?
B?
B? Okay.
Yes. I'm the one using iPhone 12. I switch
No, that's okay.
because I'm going for my third class.
What's your name? What's your name? So I don't.
Tohib.
Tohib. Oh, yeah, okay. Yeah, this is you. Okay, okay. You just changed your phone. I see. All right. So I got some B's. I got some C's. All right. Let's see what's going on here. This is the outer membrane. This is the inner membrane, right? And by now you should be familiar with the story. The ETC will pump H plus into the intermembrane space, right? Okay, so this is intermembrane space. Now the HCN, the hydrogen cyanide, will bind to the ETC. It tells you right here in the question, and it will block the ETC. So we are no longer pumping H plus into the intermembrane space. If you're not allowed to pump H plus into the intermembrane space, you would have less H plus, right? less H plus, right? Because the cyanide blocks you, right? So with less H plus, what's going to happen to your pH? Less H plus means higher pH, right? It says low pH is caused by higher H plus. So low H plus is higher pH. Does that make sense? Okay. So please be careful on the quiz, on the test. Read the question. This is asking about intermembrane space, right? Like if I ask you about the matrix, then it's the other way around, right? Okay. All right, think about that. Last part here, we're gonna go and talk about fermentation. So in humans, when we are doing strenuous exercise, like think about running at full speed, um, you won't be able to keep that up forever. You may be able to run at full speed for like 1 minute, 2 minutes, 3 minutes, maybe even, okay? But eventually you're gonna reach a point where you will feel the burn in your legs and you just have no choice but to stop, right? So when you're feeling the burn, your body is basically telling you that the oxygen demand is exceeding the oxygen supply. Right? You're not getting enough oxygen into those legs. That's what it means, right? Okay, and then what do? Well, we switch to lactic acid fermentation. Okay. As I have been trying to, drill in your head for the past half an hour, when you don't have enough oxygen, you cannot pull the electron away from the ETC. And that clogs up the ETC, right? When the NADH comes, it will have nowhere to put the electron. And that stops the whole ATP production, right? The purpose of lactic acid fermentation is to provide an alternate destination for the NADH, okay, for the electron to get off. So instead of going to the ETC, which is clogged at the moment because we don't have enough oxygen, you will just give the electron to the pyruvate, and that will convert it into lactate. So that regenerates the NAD plus, which allows you to go back and let glycolysis just keep going. Right? So glycolysis doesn't create a lot of energy. It creates only 2 ATP, but it's better than nothing. Right? That extra 2 ATP is going to allow you to go on a little longer, right? Maybe extra, half a minute or so. Right? But eventually, you will completely stop your muscle. You don't have enough ATP to go on. Right? And you will have no choice but to stop. So that's lactic acid fermentation. So after working out really hard, sometimes you feel sore for the next couple of days, right? That's the lactic acid that's building up. That's one of the causes of those burns. That just shows you the diagram of what happens. Fermentation also happens in microorganisms. Right? There are some organisms like yeast that are missing the mitochondria. But they are quite small, right? They're microscopic, and they don't need to have as high of a demand for ATP, right? So they don't have the mitochondria. They are fully capable of surviving on glycolysis alone. Okay, so in their kind of fermentation, they produce carbon dioxide as well as ethanol, right? Which is why we can use yeast to make bread, right? When you add yeast to your dough, you usually keep the yeast in the fridge, right? Like I have some in the fridge. Like, but when I make a bread or pizza, you take them out and then you activate them. Okay, when it gets warm, they start to divide. They start to metabolize, and they create carbon dioxide, which is what causes the dough to rise. And the alcoholic fermentation will also create ethanol, right? So that is the drinking alcohol. There are different kinds of alcohol, right? Not all kinds of alcohol can be consumed. Like the one that you clean your tables with or your phone with, that's that's isopropanol. That you cannot drink that. You die if you drink that. There's methanol, for example, also not for consumption. So only ethanol is the drinkable kind, and that's the result of alcoholic fermentation. So you sometimes need to ferment things, right, to produce the alcohol. There's a lot of fermented product that use yeast, right? Things like miso, right? That's fermented. Um, and then some chili paste, right? They also are fermented, and it basically make use of this process here. Again, that's just a summary. You don't have to memorize it. So basically, there are 3 possible fates for pyruvate. The main one here in the middle, that's the one we spend a lot of time talking about. Right? We got the pyruvate becoming the acetyl-CoA, right? The 3 steps. Carbon dioxide is released, we capture the electrons using NADH, and then we add the coenzyme A to get acetyl-CoA. That then proceeds to citric acid cycle and oxidative phosphorylation. That's the main one for humans, right? When we don't have enough oxygen because we are doing strenuous exercise, then our body switches to lactic acid fermentation. It allows the NAD plus to regenerate, right? So we keep glycolysis going for a little bit longer. It's not a lot, it's 2 ATP, but like I said, it's better than nothing. It'll keep you going a little longer. Right? And then you will have to stop, right? And get oxygen back, and then you can switch back to the aerobic. Right? Aerobic means needing oxygen, right? And this can happen without oxygen. For things like yeast or simpler microorganisms, they don't have the machinery to do Krebs cycle and all that stuff. So they rely on alcoholic fermentation all the time, basically. Right? And they create carbon dioxide as a byproduct, as well as ethanol. Okay. And that's it. You finish half of the course. Congratulations. Right? Your test is going to be next Thursday. No class next Thursday. The test will be covering lecture 1, 2, 3, 4, 5, and the lab as well. Okay, the beetroot lab. I returned the beetroot lab to you already. I left some comments in some of your lab reports. You can take a look at that. It's in the Dropbox where you submitted it. I will post a review sheet for the test after class. You can start working
Excuse,
on it.
excuse me,
Yeah,
sir.
sure.
Is it that we are doing the lab in the test?
No, I said the lab will be, there will be questions about the lab on the test. That's what I mean.
Okay.
Okay. Not a lot, just like 4 or 5. Okay. Where was I? What was I saying? What did I say? Yes, there will be a test review package that I'll post after class. You can start working on it. We will take it up next Tuesday in class. But start working on it. Okay, that's gonna help prepare you for the test. I'll post this information as well, but the test will be 44 multiple choice questions. Okay, 44 multiple choice questions plus 10 multi-select questions. Okay, so a total of 54 questions. Okay, 10 of them will have more than one answers, and then 44 will be just regular ones. No short answer this time. You don't have to type anything. Some of you might be happy about that. And it will be 100 minutes. Okay, 100 minutes. So that's like an hour and 20 minutes. Okay, should be plenty of time. Any questions for me before you go?